What we're seeing at ASCO 2019 is ongoing evidence that anti-CD19 CAR T cells are truly transformational therapy across all B-cell lymphomas. And one thing that we're seeing nicely here is that there is evidence now of activity of these CAR T cell products in diseases outside of where they've been initially FDA approved. So while initially we know that anti-CD19 CAR T cells are truly transformational treatment for patients with relapsed and refractory diffuse large B-cell lymphomas, we're seeing more data at this meeting for patients who are typically not included in those standard trials. And what I mean by that are patients with secondary CNS involvement by their large cell lymphoma or alternate histologies, including mantle cell lymphoma and chronic lymphocytic leukemia. Well, it's become a complicated question with now 2 FDA-approved CAR T cells in the United States, multiple more in clinical trials. How do you choose the best product for a given patient? And I think one essential message is all CAR T cells are different from one another. They are not all the same drug. So you can't take what you learn from one CAR T cell and apply it to all of the others and expect the same results. So for example, we have axicabtagene ciloleucel, which is the first product FDA approved for diffuse large B-cell lymphoma. And it's extremely effective. We see responses in just over 80% of patients and durable remissions even 2 years later in about 40% of patients. We also see that patients with refractory DLBCL can reliably receive that product turned around and manufactured in less than 3 weeks. So these are great news for axicabtagene ciloleucel. So what's on the other end of that? Toxicity. And axicabtagene ciloleucel does appear to be the most toxic of the currently available FDA-approved CAR T cells. The cytokine release syndrome rate is over 90%. 2/3 of patients develop neurologic toxicities, which can be severe, and a high proportion of patients need rescue medications, including tocilizumab and steroids. And so this product might be more difficult for an elderly patient or a frail patient. On the other end of that, we have tisagenlecleucel and lisocabtagene maraleucel. Now, unlike axicabtagene ciloleucel, which uses a CD28 co-stimulation domain, the other 2 products use a 4-1BB co-stimulation domain. Why is that important? Well, CD28 co-stimulated T cells lead to a very rapid, robust, and peak expansion early on, and that might lead to a higher peak early incidence of toxicity. The 2 4-1BB CAR T cell products are a bit more gradual, and that blunts the degree of toxicity that we're seeing. Now, both tisagenlecleucel and lisocabtagene maraleucel also induce high response rates with durable remissions of about 40% at 6 to 12 months. So I'd say the efficacy across all 3 products is looking quite similar. The axicabtagene product is definitely more toxic than the lisocabtagene and tisagenlecleucel, and so patient selection means thinking not only about the need for efficacy, but the need to get patients through the treatment safely and comfortably. I will say that axicabtagene gets the product to the patient the quickest, and in relapsed refractory DLBCL, oftentimes time is of the essence. And so for my youngest, sickest patients, I absolutely reach for axi-cel for those patients. The other thing we're seeing at this meeting is use of diffuse large B-cell lymphoma patients with secondary CNS involvement and treatment with CAR T cells. Now, previously there has been no data whatsoever telling us that we can use a CAR T cell to treat a patient with disease in their central nervous system. In fact, these patients have been excluded from the clinical trials of both axicabtagene ciloleucel and tisagenlecleucel. Our study of lisocabtagene maraleucel allows treatment of patients with secondary CNS involvement as long as they also have measurable systemic disease. So far, we've treated 6 diffuse large B-cell lymphoma patients who've had involvement of their central nervous system at the time of their CAR T cell treatment initially. And among those 6 patients, 4 of them have had a complete remission, both in the brain and in their systemic disease. 3 of those have been sustained out beyond 1 year, and 2 of these 6 patients continue in ongoing remission now well beyond 1 year. So what we're seeing importantly is evidence that CAR T cells do get into the brain and the spinal fluid. They can successfully eradicate diffuse large B-cell lymphoma either in the brain or the spinal fluid, and these patients can have durable remissions without an increase in either neurologic toxicities or cytokine release syndrome. The other exciting data we're seeing is in mantle cell lymphoma, and we've thus far treated 17 patients with mantle cell lymphoma in our study. Among those 17 patients, 12 have achieved have achieved a remission. 9 of them have achieved a complete remission. Many of those are ongoing and durable remissions with a similar toxicity profile as we've observed in the overall study. We've also seen data now with our CLL study showing that the majority of patients treated with lisocabtagene maraleucel have not only achieved a response, but have achieved MRD undetectability by 30 days after their CAR T-cell infusion. So now at this meeting, building on our data in relapsed refractory DLBCL, we're seeing that lisocabtagene maraleucel has activity and an effective safety profile in CNS involvement by diffuse large B-cell lymphoma, in mantle cell lymphoma, and in high-risk chronic lymphocytic leukemia, which has already failed BTK inhibition.